I'll hop right into introducing Dr. Stephen Djordjevic. He's a distinguished professor of infectious disease in the Australian Institute for Microbiology and Infection at the University of Technology, Sydney. His research interests are in antimicrobial resistance and pathogen evolution through a One Health lens. His laboratory conducts genomic epidemiological analysis of a broad range of gram-negative bacterial pathogens and the mobile elements they carry from hospital and veterinary and clinical environments, food production animals, impacted wildlife, wastewater, and natural water systems. So without further ado...
So firstly, thanks to the organising committee for giving me this opportunity to speak. I'm going to be talking about antimicrobial resistance and pathogen evolution from a One Health context using E. coli as a model organism, which I am very biased in thinking is one of the best One Health organisms to study. So E. coli is a minor but important member of the gut microbiome. It's a constituent of the microbiomes of mammals, birds, reptiles and fish. It's also found in soil and water, colonises plants and contaminates foods. It's excreted at between 10 to the 6 to 10 to the 9 organisms per gram of faeces, depending on the host. And importantly, it's a facultative anaerobe, which means it can grow both in oxygen and out of oxygen. And it's one of the first microorganisms to colonise the gastrointestinal tract immediately after birth. So almost everyone carries E. coli, and you carry it for life, whereby it plays a very important part in removing the oxygen content out of the gut and creating a favourable microenvironment for the establishment of a healthy fecal microbiome and gut microbiome. So in order to do that, it needs to have what are collectively termed as virulence-associated genes, but these are more probably akin to fitness factors that enable the E. coli to effectively colonise and colonise all the available sites in the gut with other microbiome to prevent the establishment of adverse microba. So apart from it being an essential commensal, it is also, and this is an important piece of information for the so what argument, E. coli is the #1 bacterial pathogen in humans, responsible for deaths associated with antimicrobial resistance. So why is that so? So the production of faeces is estimated in a 2014 study in Nature Sustainability at about 4 by 10 to the 12 kilograms per year. And it's increasing at a significant rate, estimated to be 4.6 by 10 to the 12 by 2030. That is substantially, there is a substantial contribution, almost 4 to 1, in fact more than 4 to 1, by intensive animal production animals and other animals. So the ratios of animal faeces to human faeces continues to increase from 4.2 in 2003 to 5 to 1 in 2014 to 6 to 1 in 2030. And it's important to remember that low-income populations bear the greatest burden of on-site faeces, mostly from animals in or near domestic environment. So E. coli is a very diverse species. Apart from it being a commensal, there are also the intestinal pathogenic E. coli and the extraintestinal pathogenic E. coli. And I'm glad that Lance Price spoke this morning, so some of these terms have be quite familiar to you. But the intestinal pathogenic E. coli are normally broken up into defined pathovars, depending on which major virulence attribute they carry. Whereas the extraintestinal pathogenic E. coli are arguably more threatening and more diverse, and I'll explain why. So firstly, the intestinal pathogenic E. coli usually contain more frank pathogens. By a frank pathogen is if you acquire an O157, you're most likely to have disease within a short period of time. Whereas if you acquire an extraintestinal pathogenic E. coli, you will have a substantial, an undefined period before you may or may not get disease. And because they are colonising opportunistic pathogens, the genomic epidemiology, or any epidemiology for tracking those infections back to source are complicated because of the time that occurs. So unless you are immunocompromised, unless you have a high carriage rate of urinary tract infections, unless you have a trauma or you have cancer or diabetes and you're immunocompromised, these organisms may never cause disease in you, even though you carry them in your gut all the time. So it's very important to understand this concept of hybrid pathogens, because these pathogen designations are rapidly deteriorating. The more genome sequences we sequence, we realise that E. coli shares key virulence factors between these pathovar designations, and if you put keywords hybrid E. coli pathotypes into PubMed, you can see that more and more people are beginning to realise this by the number of publications. And these hybrid pathotypes are extremely difficult to detect from a clinical perspective, and they have a history of causing devastating consequences, which I'll talk a little bit about later. Okay, so E. coli causes both intestinal infections through the IPEC and extraintestinal infections, which include urinary tract infections, bloodstream infections. In fact, they're the leading cause of bloodstream infections, full stop. The second most frequent cause of meningitis. They're the leading cause of ventilator-associated pneumonia in hospitals, but most people don't know that. They also cause significant kidney infections, as well as prostate infections. So when you think of E. coli, just don't think about diarrhoea and diarrhoea-associated infections. So for an organism that's touted as being the most intensively studied in the world, there are enormous knowledge gaps, particularly around the ecology of E. coli. So I asked a postdoc in my group to pull down the designation of E. coli sequence types since the advent of Enterobase. So in 2015, there were just over 2,100 E. coli sequence types, and by 2023, there was 9,000 E. coli sequence types. So we are adding anywhere between 800 and 1,200 E. coli sequence types annually. And you can see from that curve that is an upward trend, which means we have so much more E. coli to understand, particularly E. coli from non-pathogenic sources. So this leads to this massive E. coli database bias, which was mentioned in an earlier talk this morning. Most E. coli genome sequences that we've sequenced are pathogenic E. coli. The databases are also increasingly biased by selection practices, particularly selection for ESBLs, whereas there is a complete dearth of commensal E. coli sequences from humans, food animals, impacted and urban wildlife, to name a few. So what? Okay, so the so what is demonstrated amply in this wonderful publication, which was published by Eric Denemer and colleagues in France. And I like this study because it points to gaping knowledge gaps that we need to be better understanding. So it looked at commensal strain populations of E. coli in community subjects in Paris over a 30-year period. And the two take-home messages from that study is that there is a parallel and linked increase in the frequency of phylogroup B2 strains from 9.4% in 1980 up to 34% in 2010. Now, phylogroup B2, if you remember the talk this morning, is the phylogroup where most of the pandemic XPEC lineages associate with. This is the most heavily virulence-laden phylogroup of E. coli. So that is a concern. Also, antibiotic resistance increased from 22.6% of strains resistant to at least one antibiotic in 1980 to 44.8% in 2010. So what that's basically showing, and I would assume that the data that comes from Paris would not be any different from data from any other developed country, and probably likely to be even more significant in LMICs and LICs, is that our commensal E. coli flora is shifting from the commensal phylogroups A and B1 to the XPEC-dominated pandemic lineages, which so the virulence gene cargo and the antimicrobial resistance gene cargo is substantially increasing over time, which means all of us are likely to be colonised by XPEC, which can cause quite significant issues. So everyone's talking about antimicrobial resistance, and rightfully so, but I always find it difficult to divorce antimicrobial resistance from virulence because they share many common characteristics. Both processes are necessary for bacteria to survive under adverse conditions, to adapt to and to survive in competitive and demanding environments, and to occupy a new niche. They are mobilised predominantly by horizontal gene transfer, and the two major bottlenecks that influence their presence is immune defence systems and antibiotic selection pressure. So, but perhaps they evolved over different timescales. So if E. coli colonises the gut and then is released into the environment on a regular basis, it's constantly cycling between diverse temperatures, it's taken up by intermediate hosts, it's mobilised around by bird species, so it's constantly having to recolonise diverse gastrointestinal tracts. And there are obviously going to be lineages that do that really well and lineages that don't do it well. So you'll have niche-adapted lineages, host-adapted lineages, and also very broad host-range lineages. So antimicrobial resistance, even though it's ancient, there's an unprecedented release of antimicrobials into the environment post-1940s. And so concomitantly with a massive increase in human population, which you can see there with that red bar here, over the last couple of hundred years. And also we've gone from 2.2 billion people in 1960 to 8 billion people in 2022. And that's going to continue to increase until the middle of this century. So concomitant with a huge human population, we also have massive animal production to maintain food for that burgeoning population, which means animal fecal populations and human fecal population productions are going to be increased substantially. Okay, so anecdotally, what does this mean? So in 1990s, human clinical infections caused by XPEC were particularly sensitive, were sensitive to most antibiotic classes, particularly fluoroquinolones and third-generation cephalosporins. That changed rapidly in the 2000s. And because of the increase in resistance to ESBLs and fluoroquinolones, there is increased reliance on carbapenem antibiotics, and you can see in that panel there that worldwide the carbapenem resistance is being reported at an ever-increasing rate. So what's the sort of work that we've been doing in Australia? So we've, as part of the Australian Pathogen Genomics program, we are looking at investigating, I've collated all the Australian genome sequences. There's nearly 5,700 of them, which we contributed from our own sequencing collaborations and also those that are in public databases. They represent a 36-year period from 1986 to 2022 from diverse sources. And the overarching take-home message is that there's a lot of phylogenetic diversity, 847 sequence types, of which about 10% are novel sequence types. So what are we doing with that data? Well, we're looking at E. coli transmission networks, trying to understand how E. coli traverses diverse hosts and environments. If so, what are the lineages that do that? What are their virulence associations? And what's the AMR resistance story associated with that? But importantly, if we have an outbreak of any organism in a clinical environment, there are very defined SNP cutoffs to define an outbreak, and that's usually in the vicinity of 15 to 25 SNPs. So if you have a clonal outbreak in a ward, you'll have no more than 25 SNPs. But from a One Health perspective, it's far more difficult to define those SNP cutoffs, and that's something we've spent a lot of time doing, and we're getting quite close now to understanding what we think is a meaningful One Health linkage. We're also studying the plasmidome in E. coli, trying to understand which related plasmids, how closely related plasmids move across these environments. If so, which plasmids are doing it, what's their virulence and antimicrobial resistance cargo, and how closely related are such plasmids. But the jewel in the crown is this 12-month snapshot study, which we're about a third of the way through. And here we are looking at trying to collate upwards of 10,000 E. coli genomes from a geographically defined region in Melbourne, looking at human clinical and importantly commensal isolates, clinical and commensal isolates from companion animals, food, extensive sampling of retail meats from public outlets, extensive collection of wildlife, also wastewater, natural water and soil environments, and also livestock, dairy and abattoir sampling, and also each of the major food production industries. Okay, so harking back now to what's happened since the 1940s and the massive use of antibiotics since then, if you look at the evolution of either emerging pandemic lineage or established pandemic lineages, you can see that post the late 1980s, this sequence type 410, which was relatively unheard of even 10, 15 years ago, has been able to acquire a massive antimicrobial resistance load, including resistance to virtually all first-generation antibiotics, all third-generation cephalosporins and carbapenems, as well as all the fluoroquinolones. And it's a true One Health pathogen. It's been isolated from humans, animals, the environment, and from all continents. We also do have it in Australia, but that data is not publicly available yet. It causes human infections, including neonatal infections, urinary tract infections, bloodstream infections, as well as rectal carriage, and it's also reported from livestock, wildlife, companion animals, and a whole range of environmental sources. So if you conduct a beast analysis, and this is some very elegant work that's been conducted in Alan McNally's group, you can see that this organism's been on the planet for a couple of hundred years. It's split up into two lineages, and one of those lineages has undergone a substantial clonal expansion, and predominantly that's happened in the 1900s, and then a massive second wave around to about the late 1980s. So you can see what massive rises in human population and animal populations do to how E. coli copes with those selection pressures. Now, ST410 is remarkable in the amount of antimicrobial resistance carriage that it has, but it's largely unremarkable in the amount of virulence carriage that it has. In fact, there is probably a whole range of new virulence attributes in ST410 that we don't fully understand how they function. But just 3 weeks ago, there's been a new clade of ST410 described in China. It's causing deaths of infants at quite a significant rate in a number of hospitals across China. And this new lineage has acquired the Yersinia high pathogenicity island, and also it's undergoing serotype switching. So this is a remarkable observation, because I would my belief is that this clone will explode across the world, because the Yersinia high pathogenicity island is probably ranked #1 as an expect virulence factor. So that's a very worrying concern. And I think people often forget about co-carriage. So here you can see the virulence gene resistance region on the F plasmids carried by ST410. You can see that in any of these pictures here, there is resistance to trimethoprim sulfonamides, a whole range of aminoglycosides, erythromycin, even ampicillin, and also then all the extended spectrum beta-lactams. There are several genes for extended spectrum beta-lactams, and it's also now acquired BLA-NDM. And you can see that this is all assembled in one region on the plasmid, and that entire region is constructed by the activity of IS-26. So IS-26 is just assembling these resistance genes, and placing them all in one region on these plasmids, and the certain different parts of these can flip in and flip out. New cargo that's flanked by IS-26 can come in. So these are very rapid systems for acquiring antimicrobial resistance cargo. The point is that if this organism finds its way into an animal production system that's very, very good with high management practices and doesn't use any fluoroquinolones or extended spectrum beta-lactams, this organism's still going to be selected for, because they will be using things like ampicillin or some first-generation antibiotics. So hybrids. Hybrids are when key virulence factors move between pathovars into other pathovars. So in 2011, we saw massive outbreak of O104H4. It caused hemolytic uremic syndrome in nearly 1,000 people. I think it was well over 850. I think there were about 60 deaths. It also caused, it was traced back to contaminated sprouts from Africa. Until they'd sorted that out, there was a lot of finger pointing, so there's massive economic losses caused by finger pointing to Spain, which then got compensation for that outbreak. And this is simply an interaggregative E. coli that acquired a chicken toxin from a from a ruminant reservoir, most likely bovine. The second one that I draw your attention to is, which hasn't got a lot of headlines, but is the ST301 outbreaks that have been occurring across Europe, particularly in France. It's the second most frequent cause of hemolytic uremic syndrome. This is an enterohemorrhagic E. coli that's acquired a Colvi plasmid. So we heard about Colvi plasmids this morning. They are certainly well entrenched within the poultry reservoir. And we know we've been doing a lot of work on how the acquisition of Colvi drives pathogen emergence. And we recently published a paper on how ST58, a sublineage of ST58, had acquired a Colvi plasmid and is now increasingly becoming a pandemic lineage. So these are the sort of frightening things that are happening all the time. Okay, so this is not my work, but certainly some very important work that's been coming out of a number of laboratories, but particularly from Alan McNally's group. So what makes certain clones or lineages of E. coli successful multiple drug resistance pathogens? So there's a couple of pieces of information to suggest that it's not the acquisition of multiple drug resistance which drives clonal expansion, although it does have an important role in clonal expansion, but rather it's the ability of an organism to successfully colonise and displace the natural microbiome. So in Norway, clade A ST131, which is a clade that's not so dominant in many other places around the world in humans, colonised a lot of people. It was pan-sensitive at that time, and it displaced, and a lot of people got colonised by ST131 and came down with illness, but then it soon rapidly became multiple drug resistant due to selection pressure. So it's the ability of the multiple drug resistant E. coli to rapidly and asymptomatically colonise the intestinal tract of humans that sets them apart from other E. coli lineages. This was also amply demonstrated in the COMBAT study, whereby they sampled travellers in their home state, then sent them abroad and sampled them intensively, and then brought them back and sampled them intensively. And it was very clearly to show that acquisition of multi-drug resistant clones from hotspot regions of the world can colonise you when you come back to your home and remain colonised for anywhere up to 12 months. So that's a concern. So AMR carriage is not equal across the E. coli population, with AMR being concentrated in certain lineages. And multiple drug resistance plasmids are not evenly distributed amongst expect lineages, but rather their carriage is concentrated in a small number of highly successful clones. And that's what we need to get better at and to try to incorporate into our risk profiling. So metabolic diversity appears to be a very key feature that makes certain clones and lineages of E. coli successful multiple drug resistance plasmids. Multiple drug resistant strain ST131s can readily colonise new hosts, even when these hosts are pre-colonised with commensal E. coli. They can even outcompete drug-sensitive, very successful pandemic lineage ST73. So it suggests that the greater carbohydrate and metabolic capacity you have, the greater capacity you have to reshape the microbiome and colonise. So multiple drug-resistant lineages normally have a greater capacity to metabolize diverse carbohydrates. And that's something that we now need to build into our pipelines to try and rank and understand this a lot better. Okay, how much time have I got left? 1 minute. Okay. All right, so I might just skip this. So in summary, genomic surveillance to date has largely been reactive. We need to get better at predicting and detecting emerging pathogens, expand studies to diverse hosts and understand commensal populations so that we start balancing our E. coli database biases. We need to monitor emerging E. coli hybrid lineage evolution and to characterise the mobilome better. We need greater assistance to LMICs and to LICs. The learnings applicable to E. coli are readily applicable to other escapee pathogens that are also important colonising opportunistic pathogens such as Klebsiella. We need to establish and improve upon the circular economy to get better at treating feces and to get resource allocations back from that. And whole genome sequencing is invaluable for monitoring the success of mitigation practices. And with that, I'd like to acknowledge the current members of my group as well as past members and for the funding streams that I've had. So thank you very much for your time.
Thanks, Stephen. I was wondering, you often see on some of these plasmids with multiple resistance genes, genes that basically encode for similar, or at least what we see as similar functions, like you mentioned beta-lactamases. So I was wondering if you have any hypothesis of why they'd accumulate genes that seem to be doing more or less the same thing.
So what I think is happening is that once you get incorporated into a multiple drug resistance region, you have very minimal fitness impost. So the organisms tend not to, they tend to represent their AMR evolution within their multiple drug resistance regions, so they don't shed those genes. Why is that so? I just think they find themselves in such diverse hosts because they're constantly passed out into the environment that having those first generation antibiotic resistances are still useful for a whole range of things where those selection pressures evolve, such as in diverse animal production systems, probably in aquaculture and various other systems that we have very poor information on.
Excellent talk, thanks. In the program that you presented, you focused on plasmidome, as most of us do, because it's easier. But is there an intention to include, more of the diversity of MGEs that are carrying either virulence genes or antibiotic-resistant genes, like ICEs, IMEs, and all the other types of mobile genetic elements that are less easy from a bioinformatic point of view to include in such studies?
I think we need to get, which we're trying to develop a sort of a ranking system for mobile genetic elements in terms of importance for virulence cargo, and also you've got to be cognizant of the fact of which host you're focusing that ranking on. But certainly ion acquisition systems are extremely important in E. coli for colonising extraintestinal sites. So you'll find that E. coli has multiple ion acquisition systems over and above the ion acquisitions that are present in commensal E. coli. So this additive effect seems to be really important. And also, there are also some lineages of E. coli where the virulence is not necessarily plasmid associated, but is associated very strongly with genomic islands, such as ST73. So ST73 hardly has any F plasmids, but it has a lot of entrenched virulence encoded within genomic islands. And so there are a number of very important genomic islands that we also track.
So, as you show in some of your data, E. coli carry antibiotic-resistant genes, for example, for azithromycin, which is not a target for E. coli. So it's prescribed for upper respiratory infections. And so every time that antibiotics are prescribed, E. coli needs to survive those antibiotics. Yep. And so based on your observations, do you think that E. coli is a reservoir of antibiotic-resistant genes, and these genes are in commensal flora more than in pathogenic flora?
Yeah, that certainly makes sense, but it's too hard to interrogate because we don't have enough commensal E. coli genome sequence data, but that would make a lot of sense. And also Klebsiella is also very, very good at doing that. Okay, both of them colonising opportunistic pathogens.
right, thanks for the introduction. So, smoking. Think you're not going to be surprised if I tell you it's probably not good for you. But where does AMR come into the equation? Well, if it's not good for your health, you're probably more prone to be getting bacterial infections, which means smokers have a higher risk of being prescribed antibiotics. So that would be a direct effect of the cigarettes. But we wanted to answer, is there actually something like an indirect effect as well? So does smoking potentially lower the efficiency of antibiotics by promoting AMR? And why is that? Well, cigarette smoke contains around 7,000 different chemicals, a lot of them been shown to be cancerogenic or purely cytotoxic to all kinds of organisms. At the same time, there's millions and millions of people smoking, and there's billions cigarette butts that are constantly thrown away, and they have a lot of entrapped toxicants. And estimates say that around 40% of these cigarette butts that are smoked worldwide every year are ending up in our waterways. So quite an emerging pollutant, and they can swim on the surface and travel quite far. So what we want to do in this study is to look, do toxic cigarette compounds co-select, trigger bacterial stress responses, promote plasma transfer, either in the inhaled smoke in the lung or through discarded filters and ash in the environment. And obviously, because it was not really ethical to employ a lot of students to smoke all these cigarettes, we created a little lab smoking station that sounds a lot fancier than it actually is. It's basically a pump, a tube, and a little bit of artificial lung mucus medium. And we smoke. The smoke goes through the mucus medium, gets collected in there, so we get some cigarette smoke condensate. Then we collect the filter and the ash and create, just in sodium chloride solution, cigarette filter leachate and cigarette ash leachate. Then we do very simple plasmid transfer assays. So we have a donor, Pseudomonas putida, that has a multi-drug resistance plasmid, PKJK5, and we have a recipient, Pseudomonas putida, that is chromosomally resistant to rifampicin. So we can, after the assay, easily do selective plating to count out, okay, how often did the plasmid transfer? And we perform that either in artificial lung mucus medium, which is like a weird mixture of like various things that make it not very liquid anymore. And we also do it in sterile river water. So in the mucus medium, we add the cigarette smoke condensate. In the river water, we add ash and filter leachate. So let's take a look what happened first in the artificial lung. Basically, we added amounts of cigarette smoke condensate to different degrees. You see it here as cigarette equivalents per milliliter. And you see, okay, there's not really any difference in growth. So it doesn't inhibit the growth of the both strains, of the recipient or the donors. But if we look at the amount of transconjugates that are built, very clear concentration-dependent effects. So why did that happen? Well, we did a little bit of an assay of, okay, are these bacteria stressed? Stress is very well known that it promotes gene transfer, and there is a clear increase for all the concentrations compared to the control in ROS that's created. So next we looked at the ash in the river medium. Only big difference is, well, there's less nutrients in there, so the numbers are lower in general, but we also see this clear concentration-dependent effect and even further amounts of stress, potentially due to all of the heavy metals that are still in the ash. We thought, okay, 2 out of 3 done, you know science. Third one usually doesn't work. So in the cigarette filter leachate, nothing really happened. First thing we thought, okay, what's happening here? But there was also some visual cues already. The ash leachate, the cigarette smoke condensate, they looked very dark and had like some coloration, while from the filters, no real color was released. And after we did some chemical analysis, there was also not much in that liquid. So we thought, okay, so if all these toxicants are staying on the filters, could these filter-bound toxic compounds provide breeding grounds for bacterial communities of high levels of risk in the environment? And to do that, my students created a lot of these little cigarette butts, either used or unused. You see them very nicely that there's definitely toxicants on there. We dumped them into a sewage stream for 5 weeks. Afterwards, took them out, analyzed them. First went to Resistomap to analyze all the ARGs, MGs, and so on through high-throughput qPCR. And then we did some 16S RNA-based sequencing. So let's take a look at the communities first. So on a lot of the next graph, you always see the unused filters in red and the used filters in blue. First thing we saw, okay, there's distinct communities on these two types of filters. And on those filters where we have toxic compounds bound and they are probably distributed rather heterogeneous, there is quite a bit more of diversity of the communities that colonize these filters. Then let's take a look at the communities. What do they have in ARG content? I'll show you here a graph for like, I think, 10 ARGs. And you see constantly for all of them, we see significantly more ARGs. And we had in total a set of like, I think, 27 ARGs. And for 25 out of those, we saw this very clear increase in these communities. Well, we thought, okay, that makes sense because all the toxicants that were in the smoke were able to promote horizontal gene transfer. So we also looked at different MGEs. All these MGEs also very clearly upregulated in the used communities. And the one thing I probably would like to say here is this is not an effect of bacterial densities on these filters. So bacterial densities were very, very similar between used and unused filters. It's like similar cell numbers. And so it's not because, okay, there's a lot more bacteria and they're more close, or they can probably utilize some of the toxic compounds as carbon sources. It's really an effect of either the colonizers have more MGEs or they transfer more MGEs. We then went to a recently published list of all of the known genera among the bacterial groups that have human pathogens among them. And we looked which percentage of our sequence communities are belonging to these genera of at least potentially pathogenic groups. And we saw, again, a very clear increase between our red unused ones at like around 15% of our sequences were belonging to potentially pathogenic genera, and then went up to around 22 to 25%. But, well, what does that mean if there's this potential? And we really want to test, okay, is there actually some kind of effect? So do we select for something that is actually pathogenic? And so we went to this lovely little model, which is Galleria malonella, the wax mouse larvae. And it's a very good model for pathogenicity because it kind of has structural and functional similar immune systems to mammals. That is, number one, because the skin is like kind of a cuticle barrier, which is very similar to mammalian skin, and it has a very nice hemolymph and hemocytes, which is similar to the immune system in the human blood. You see them here. They are like around, I don't know, 3 centimeters long, and one of my students had to get like 1,000 of them. We extracted the communities from the filters, measured, okay, how many bacteria are in there so we could normalize bacterial numbers, and we injected them at the hind leg of these little wax mouse larvae, and then looked into, okay, if they're pathogenic, we should see an effect, and the effect usually looks like this. So they turn completely black and they melanize, which makes it very easy to judge, okay, they're dead now. They don't move and they're black. Sounds good to me. So we inserted the communities from our used and unused filters, and if we look at that, you see here the percent of survival. Oh, I just see up here it's supposed to say 100%, and down here it's 0%. So we have, I think this is around 100 of these wax mouse larvae that were injected with the communities from different filters for each of the curves. And you see here, the unused Marlboro Reds die after like around 20 hours, so there is also something that's pathogenic, but a less slower proportion. And around 15 hours, we see the death already in the used filter, so a lot more pathogenicity on there. We then thought, okay, there's different kinds of cigarette butts. What might happen if we use differently strong cigarettes? So we've got light cigarettes, medium cigarettes, heavy cigarettes. Do we see any differences? To our surprise, what we saw is the ones that died the earliest were these blue ones, which were the ultimately light cigarettes. So if they're so light, why do they kill earlier? So we went a little bit into the literature about like these cigarettes and how they are made light. It's actually the exact same tobacco, and it's just that they use better filters that trap more toxicants. So there's more toxicants in there. They select heavier for these pathogenic AMR, mobile genetic element strains, and suddenly we say, okay, lighter cigarettes worse for the environment. We're now also looking into if we spike our Galeria together with the community and antibiotics, do we see effects of this increased antibiotic resistance? But sadly, I don't have any curves for you with that. But with that said, I have a lovely picture for you as my conclusion slide. Cigarette consumption promotes the spread of AMR. We have, number one, these dangerous hitchhikers on our cigarette butts that can be transported far to novel ecosystems. They can be transported over 1,000 kilometers, have been shown for certain cigarette filters. They get eaten by seabirds and so on. Kids at the beaches might play with them, so we don't want them, and they bring all of this cargo of ARGs, MGEs, and virulence factors. We don't like that. At the same time, all of the smoke that's consumed is increasing plasmid transfer in the lung. So maybe as the final conclusion, if you don't smoke, don't start. And if you smoke, probably take care that your filters don't end up in the waterways. With that said, I would like to thank especially my 2 PhD students who had to work in a lab where it smelled like smoke for quite a time. They had to inject all of these little larvae, so maybe not the most favorable task to do, but they actually quite enjoyed it. That's Pei Zhu Fang, who just graduated last month, and Diala Konyali. At the same time, I'd like to thank all of the collaborators, some of which are here in the room, the funders, the projects, and if you want to have a look at the preprint, which is not including yet the Galeria experiment, then please scan this code. And with that, I'm done. And if you have any questions, please do.
Tim Vogel from the University of Lyon. Very interesting. I'm wondering about, there's several reports about the microorganisms on tobacco leaves and cigarettes and possible pathogens in those communities. I'm wondering how they may affect what's going on in the filter or not. Do you have any ideas about that?
Well, I would assume that rather than the things on the tobacco leaves, because, well, they get heated up so much during smoking that they probably die. It's probably easier coming from the other side of the filter, so human mouth bacteria that in addition are colonizing and have potentially priority effects. Similarly, if they are not completely smoked, there might be effects, but I think the tobacco is usually reasonably dry enough that a lot of the bacteria will probably not survive until there.
Yeah, I think the group in Johns Hopkins showed some survival into human lungs, but I don't know the details of their work. But
I'll have a look at that. Yeah. Sounds good.
Concepcion Sanchez from the University of Lyon too. I have a couple of questions somehow related. So the first one is concerning your percentage of genera, including pathogenic, potentially pathogenic strains. Do you think that's just because so you're breathing into the filter and then they're human associated, and so those genera are more associated potentially with pathogenicity? And then related to that, so when you see a higher abundance of mobile genetic element associated genes, I'm wondering about their content. So is this increased abundance because the bacteria that carry them are being promoted in the filters, or are there any genetic changes going on inside the mobile genetic elements, or an increased transfer? Do you have any information on that, or any plans on looking into that? Thank you.
So the first question is reasonably simple to answer, because, well, we used the smoking robot, so there were no human-associated bacteria on the used filters. And in addition, yeah, we don't really know if it is pathogens that are preferentially, well, we think it's pathogens that are preferentially colonizing, but we don't know if the MGEs are transferring or if it's just that, in general, potentially pathogens are used to adverse environments and might be equipped with mobile genetic elements and are going on there. So that's a little bit questionable, but right at that very moment, now one of my students is cutting these filters into tiny slices, injecting them with single strains of like these plasmid transfer things, and performing it on the filters, as also one of the reviewers had the similar question that you had, and we're now trying to prove exactly that. So is it, or what are the rates of plasmid transfer we can expect on there, and
Thank you for lovely introductions, and also thank you for EDA to give me this wonderful opportunity to talk about my research. My talk may be a bit more serious and less exciting than the previous one. So I'm going to talk about the impact of microplastics on bacterial community and antimicrobial resistant genes in Norwegian and South African wastewater. As the antibiotic resistant rates continue to rise, we are now at the intersection. On one hand, we continue our race to discover new antibiotics, especially in the era of AI, develop intervention strategy using microbiota or vaccines, explore alternative treatment such as phage and antibody therapy. On the other hand, we still need to focus on preventative measures in the One Health continuum. The One Health continuum considers animal, human, and the environment are interconnected. To do that, we need to identify AMR hotspot, where the source of AMR is and how it's spread into the environment. And it has been mentioned a lot in this conference, no doubt wastewater treatment plants is one of them. Wastewater contains a lot of chemical pollutants, plastics, debris, and numerous pathogens. In this project, we want to understand the role of microplastics in shaping bacterial community and how it contributes to the spread of AMR. Microplastics is defined as plastic pieces that has size smaller than 5 millimeters. So the project is actually a joint collaboration between Norway and South Africa. We selected two wastewater treatment plants. One located in Tromsø city, if you don't know it, is actually above the Arctic Circle. And the other one is Potchefstroom in a city in South Africa. This slide shows the diagram how we set up the experiment. It's very simple. Actually, we just went to the wastewater treatment plants, collect the inlet and outlet wastewater sample, brought them back to the lab and incubate in the flask containing different kind of material. After 1 week incubation, we extract DNA and then we sequence them on a MinION platform, and we also did qPCR on some gene target. And we'll see what we get. For the study, we chose 2 different kinds of rocks, the black rocks and the white rocks. Also, we order or purchase different kinds of microplastic types from the company. They are PVCA, HPDE, PLA, PET, and PE. The figure was from the previous study that they show how the structure of the surface change when the microplastics get exposed to the environment, actually to the chemicals. You can see a lot of cracks and holes on the surface, and the idea is that this structure will promote the AMR spread and support the biofilm formation better. To test this concept, we used 2 artificial aging methods. One is UV light and the other one is hydrogen peroxide. Here I just want to show you our preliminary data. When I plot them on the to see the top 20 bacterial genera from each testing material, we see that very distinct pattern in each material testing. But we also spot some overlapping common genera across the testing materials, such as Acidophorus, Chromamonas, Acinetobacter, and Acetoanaerobacterium. And next, I performed to look at the bacterial community using the full-length 16S RNA gene dataset. When I visualize the data using non-metric multi-dimensional scaling, I saw that they group really well when I group them into either plastic or rock or specific material types. But for the treatment type, it's just all over the place. And the analysis statistical testing also gave the results in line with what we visualized the data, where I got a very significant p-value for grouping the plastic or rock, grouping specific material. I also see the significant effect from the sampling date. For the treatment, it didn't show the significant p-value. And next, I performed the indicator species analysis. So basically, this analysis will tell you which species have a strong association with which material. I couldn't bore you with a long list of species names, so here I just show the summary of the analysis. And it's very interesting that we see only one indicator species that has strong association with the PE and PET group. And this isolate actually was isolated in 2010. And it's a putative human pathogen isolated from a human blood sample. And sorry, it was the inlet. I just show you the inlet samples. And next is the outlet sample from Norway. Similarly, we also see the samples cluster really well when we group in plastic and rock. Specific material, the significant p-value agree with what we see from the loss. In this setup, I didn't use UV, but I aged the material with hydrogen peroxide instead, and then I still see the significant effect from the treatment. And for the indicator species analysis, I was able to identify a list of species that has strong associations with each material testing, also with different kind of combined group. But I want to point out that we were able to identify a list of bacteria that only show strong association with the plastic, but not the rock group. And one of them I just found out yesterday from one talk, the Stenotrophomonas matophilia is the human pathogen, was among them. And we also performed the same similar analysis on inlet and outlet wastewater in South Africa. And again, we see a significant effect when we group them into specific material. Although in this time, we didn't see the significant effect when we group into plastic or rock. For the treatment, again, we only used hydrogen peroxide, and we see the significant effect when we do it on the outlet wastewater samples setup. And indicator species analysis showed that many Acinetobacter had a strong association with the plastic group, but not the rock group. We also did qPCR. We performed qPCR on two different gene targets, the Plasmox gene and the Plasmox gene. So what we see here is that the gene abundance in the outlet wastewater in Norway is significantly decreased compared to the inlet. However, we see the opposite in the South African wastewater, where the gene abundance is higher in the outlet compared to the inlet. We also perform whole metagenomic sequencing on MinION platform, but in this case, we only did it for Norwegian samples, and because it's difficult to see the whole heat map, so I cut out a small section and zoom in for that, so that you can see it. What we found is that three different kinds of plastic, PE, PET, and BLA, seem to increase some certain AMR genes, while PVCA decrease them. In conclusions, that I just show you a specific material type has a strong effect on the microbial community. Material weathering using hydrogen peroxide also seem to have the effect. We also identify a list of indicator species that represent in each testing material, and especially in the plastic group. And finally, PE, PET, BLA increased some AMR genes while PVCA decreased them. And with that, I would like to thank the project leader, Organoquimac, and our South African partner, Carlos Benstenhout. Sorry, I didn't know how to. And our funding agency, the Research Council of Norway, and the South African National Research Foundation. And yeah, thank you for your attention. I'm happy to take any questions.
Hello, my name is Paola Grenni, Water Research Institute of National Research Council in Italy. Very interesting talk. And do you know why there is these differences among the type of the plastics and the resistant genes? Do you have an idea for what?
Actually, I just look up some, like you have some explanation. And they say that PVC is normally used in the sewage pipe. And most of the studies show that it has more toxic property compared to PET and PLA. PET normally used in like commodity packaging, so it makes sense that it should not be too toxic. Yeah.
Hi. I'm Yan Ping Mao from Shenzhen University. It is a very interesting talk. My question is about the identification of the microplastics from the inlet or the outlet of the sewage treatment plant. I think it is a very challenging work for us to identifying the microplastics from the real wastewater, right? And how about the shape of the microplastic used in your study? Is it a fiber or particle or any other shapes?
So the information I have is that in South Africa, wastewater treatment plants, they only have very basic string, and the size is about a few millimeters. But in Norway, we have a very fine string, which is about 300 micrometers. But I think it's made from metal, but I'm not so certain about that information.
Okay, another small question is about, you seem that the plastifier, maybe the biofilms on the surface of the microplastics may accumulate the AMR or antibiotic resistant genes, right?
Yeah.
So have you considered what's the reason for that?
The reason is that I think is that in some plastic, it could be a food source for the bacteria to survive, to eat. Also, it provides a surface, the vector, that the bacteria can bind to and then can form like a biofilm, and yeah.
Thank you, Jay, for the introduction. Thank you for this opportunity to talk about one of my projects related to environmental dimension of antibiotic resistance. So the background of this photo is taken from Lake Erie. When there's a really heavy bloom happens, it's called harmful algal bloom, and then it looks like that. So today I'm going to talk about whether there's a relationship between harmful algal bloom and is there some potential public health impact. So before start, let me introduce my team projects. So we have 5 PIs involved, led by Dr. Soryong Che at the University of Cincinnati. So we have a quite interdisciplinary team. So I'm a microbiologist. My lab is in charge of microbial community characterization. And then Dr. Che and Dr. Dion are the ones environmental engineering, and then Dr. Dion is analytical chemist, but unfortunately, in the middle of our project, he passed away suddenly. And Dr. Helmicke and Hunt are the computer scientists. So the overarching goal of this project was providing really real-time adjustment for drinking water plants, so to prepare harmful algal bloom incidents, to provide safe water. So not preventing over treatment, but also suboptimal treatments. Because when harmful algal bloom happens, if drinking water plants to do over treatment, including like a heavier, more like oxidants, it can generate harmful algal bloom, disinfection byproducts. And then also at the same time, when harmful algal bloom happens, the drinking water has higher level, especially the source water have higher level of toxins. So the optimization in real time is important. So I'm going to talk about one of the objectives in doing this presentation. And today's presentation is mainly from one of our manuscripts that we are about to submit. The first author is Abby Volk, one of my grad students, and then also Dr. Molly Mills, my postdoc in the audience. She's going to present another poster that is about wastewater, fish gut microbiome and wastewater. And then Dr. Che is one of the authors. So we are going to talk about drinking water source. If the water source is heavily impacted by harmful algal bloom, that includes a lot of things. And then what would happen in the drinking water? But today is mainly focusing on the source water that goes into drinking water plants. So what is harmful algal bloom? That is, in fresh water, it is mostly cyanobacteria. This is not just limited to Western Lake Erie, but actually it's a global problem, including all continents in the world. So the problem is these are phototrophic bacteria, so that means they need inorganic nutrients, especially nitrogen and phosphorus. So all the eutrophication together with climate change, increasing temperature, give a really favorable condition for them to proliferate. So the thing is that under climate change and other factors, so the increasing frequency, duration, and severity are all consensus. So cyanobacteria in freshwater, especially cyanobacteria, are the problems. These are the major players, including Anabina, now it's called Dolichospermum, and then Lyngbia and others, but Plankthorax and Microcyst are the ones, two major players found in North America and China and South Korea and many other places. But usually, when we have a cyanobacteria in water, mostly the focus has been their toxins, such as microcystin. And microcystin is a liver toxin. It can cause liver disease, but also long-term chronic exposure can lead to liver cancer. But other anatoxin, toxin, they are neurotoxin. BMAA is known for linking to Alzheimer's and Parkinson's. So the question for this meeting is that whether harmful algal bloom is related to ARGs and registrum. The answer is yes, this is an emerging topic, so more literature support this idea. So we published one review paper for this, and then the idea is that, so under climate change, we have increasing extreme weather events and then increased temperature. All the conditions lead to more nutrient input in our receiving water body that can be used ultimately for food production and then also drinking water source. So when harmful algal bloom happens, cyanobacteria bloom happens, so this is a heavily, densely biofilm is formed. Usually they are aggregated, And then, so that's a kind of perfect condition for a lot of bacteria can meet together, and then their social network, promoting like a horizontal gene transfer, but also where there are cyanobacteria bloom, there are lots of phages that they're including cyanophages. So all this kind of conditions are perfect for AR spread. And then also literature says that climate change, especially increasing temperature, can promote more like a conjugation. So one paper from this group says that compared to 25 to 30 and then above 37, up to like a 10,000 times more gene transfer can happen. So another papers led by Wang's group and Dial's group also showed that ARGs detected in cyanobacteria. So all kind of genes that we also found in our study. And then also our study, not published yet, but we found that cyanobacteria were hosting ARGs and then MGs. So the question is, cyanobacteria, we found that the more kind of accumulating data point that cyanobacteria bloom can contribute to the registome and host ARGs. However, then how about drinking water sources in bloom-affected regions? So for that, here is United States, the state Ohio, where I come from. And then this is the county map. And then at the north part, we have Lake Erie, especially western Lake Erie is a heavy bloom. The city Toledo is famous for once in a while or sometimes we have a very heavy bloom. Another Salina has an inland lake, Grand Lake St. Mary. So those two are our study sites. The red dots show the public water system that detected microcystin in their source water. So Toledo is the western Lake Erie is here, and then in Toledo City, we have a lot of heavy bloom, especially in Maumee River that flows into Lake Erie. So this is just kind of regular time. We see the Salina, the drinking water intake from Grand Lake St. Mary, Toledo, the water intake from western Lake Erie. So previous studies, including our study, showed that West Lake Erie has predominantly by microcystis, and Grand Lake St. Mary is predominant by plankthorax. So for that, we got the water from Salina drinking water plants and then Toledo water intake. We got the water for 1 year, including a little bit of pre-bloom season, bloom season. We got the water, and then we tested toxin using ELISA or LC-MS. We tested microcystin and also SOX-toxin and anatoxin. But also we do metagenomic sequence, shotgun sequencing, and then also droplet discharge PCR for bacteria and ARGs. And then also for viruses, we did InnovaPrep concentration also with the shotgun. But for today's presentation, is primarily focusing on bacteria and then ARGs. So this is the one of the results, droplet discharge PCR for targeted measurements. So the result, we can see that this one, PCIGS, that detects toxin-producing and non-toxin-producing microcystis. And then MCYE is one of the microcystin-producing genes, so that means toxic microcystis. MCYE plankthorax is toxin-producing plankthorax. And then this one is SOX-toxin and anatoxin producers. So we saw that the Lake Erie and Grand Lake St. Mary, we saw that Lake Erie, Toledo has higher level of toxin-producing microcystis is predominant, and then in Grand Lake St. Mary is plankthorax. And then also our microscopic measurements also confirm the results. And Toledo has predominant by Microcystis, and Salina has filamentous plankthorax. And then also toxin results, and the Microcystin show that Grand Lake St. Mary Salina has much higher level of Microcystin in their source water, but Lake Erie is much lower, maybe because Lake Erie, we started May, and the May, it's before the bloom starts. How about ARGs measured by droplet digital PCR? So one of the genes we measure are MGE, but also we SOL1 and then TEQ. We can see that Lake Erie has significantly higher concentrations of those three genes, I mean, especially SOL1 and MGE, but TEQ was similar. So for the sake of time, I'm only showing the cyanobacteria community based on our shotgun sequencing data. So this one is average gene abundance between cyanobacteria genera. And Lake Erie is predominant by Microcystis, and then also FM isominant. So the FM isominant is one neurotoxin producer, and then also Synachococcus. Compared to that, Salina water has heavily predominant by plankthorax. And then also the beta diversity plot shows that Grand Lake St. Mary and the Lake Erie, they have a very distinctive cluster, but May, before bloom season started in Toledo water, also distinctively different. How about ARG? This is the highlight of today's presentation. So we used deep ARG and then also card RGI. So left panel shows deep ARG results. We see that Lake Erie and then Salina has a different abundance of ARG class. And then right side is card RGI. So for example, Lake Erie is there is a glycopeptide resistance, and then disinfect agents and antiseptics, but also fluorochlor, especially tetracycline. And then compared to that, Salina has a simpler composition, including glycopeptide, and then also disinfection agents and antiseptics. And then the next question is, who holds the ARGs? So for that, this one includes all the microbiome data. As we can see, actinobacteria and proteobacteria are the major ARG hosts. However, cyanobacteria, this color also quite significant hosts of ARGs. So takeaway from today's presentation is that the rhizostome generally lacks pathogens and then ARGs of clinical concern from the samples that we used for this study for 1 year. And then also one of the genes annotated, hosted by cyanobacteria are the top three are OXA, VAN, and then QHC. So OXA annotation looks like in Synachococcus that we found quite a significant proportion from Toledo water, and then that shows beta-lactamase. And QHC annotation found in plankthorax, especially from Salina. And then this one is the multi-drug resistant efflux pump. And then another one is vancomycin resistance throughout the cyanobacteria community. So when the water is heavily contaminated by cyanobacteria bloom, cyanotoxin is one thing. Also, increased organic matter is one thing. So that affects downstream treatments, increasing disinfection by products. But also ARG is another concern. But for future study, I would recommend another method of sequencing, such as long-read sequencing, But from shotgun, we kind of found a little preliminary data. So with that, I'd like to thank our group and then also funding agency. It's from Ohio Department of Higher Education, especially Humphrey Bloom Research Initiative. Thank you.
Thanks very much. Very interesting talk. I just wanted to ask about the control strategies that are used in your area for controlling algal blooms. Do you have copper treatment at all in any of the reservoirs?
So for drinking water, one is the first step is not to break the cyanobacteria cells. So during coagulation and flocculation, not to disturb those cells. So as much as they can remove. And the next step, for example, in Toledo, they installed ozonation because 2014, the finished water showed microcystin. So they invest a lot of money, so ozonation. But also during summer bloom season, they usually add extra amount of PAC, powder activated carbon, to remove any remaining toxin as absorption. So then, another disinfection. So that's for drinking water part.
That's in the treatment plant, but what about in the reservoir? Is there any use? Because in Australia we have some states that use copper and some that...
Oh yes, so copper sulfate is one of them. Yeah, that's for at the reservoir level, copper sulfate. And then in our lab, we are using UVC LED at the kind of beginning of this, the reservoir, and then like a recreational water. Thank you.